prokaryotic versus eukaryotic cells first. So eukaryotic cells are cells with a nuclear membrane and organelles. So for example, plants, animals, fungi, and protists all are eukaryotic cells. So we've got a diagram of a eukaryotic cell right down there. So we've got some of the features and some of the elements that are included in a eukaryotic cell. But essentially, we want to remember that it has a nuclear membrane and organelles. Right? When we compare that to the prokaryotic cell, it is cells with no membrane-bound organelles. So prokaryotic cells are much simpler than the eukaryotic cells, and they are mostly unicellular, for example, bacteria. So you can see the comparison. It looks a lot more simple. There's a lot less organelles even than the eukaryotic, from what you can see in the diagram. So you want to be able to identify that and then be able to differentiate between them. So one has a nuclear membrane, one has no membrane-bound organelles. Okay, so that's the difference. And you want to be able to remember examples for each one when you are differentiating. So eukaryotic, plants, animals, fungi, protists, prokaryotic, bacteria. Okay, characteristics of a eukaryotic cell. So they have membrane-bound nucleus and membrane-bound organelles, like we talked about in the previous slide, right? So this idea that most of these are going to have a membrane compared to a prokaryotic, which does not have any membrane-bound organelles. So with eukaryotic, unlike prokaryotic, which just have unicellular or are just unicellular, eukaryotic can actually either be unicellular or multicellular. So organelles perform their specific function and carry out biochemical reactions essential for cell and organism function. Right, so it's important to take that into consideration when we are talking about eukaryotic cells. So this idea that they have specific functions, they carry out those biochemical reactions, and they're essential. So therefore, DNA for the cell can be stored in the nucleus. Okay, so that is the idea of a eukaryotic cell. Now, compare that to prokaryotic cells. These do not have membrane-bound organelles like we first talked about here. So there's no membrane-bound nucleus, unlike a eukaryotic cell. There is no membrane-bound mitochondria either. DNA is stored in the cytoplasm, and non-chromosomal DNA is stored in the plasmids. Okay? So what are some examples, like we went through earlier for prokaryotic, there's bacteria, there's also archaea. So here's a bacterial DNA, the plasmids. We talk about the fact that non-chromosomal DNA is stored in these plasmids. Okay, so we've got the diagram here of the prokaryotic cell. We've got the key terms here as well, in case you were a little bit confused as to some of the terms I'm referring to. We've got organelle, which is an organized or specialized structure within the living cell. We've got membrane, which is a thin sheet of tissue or layer of cells acting as a boundary, lining, or partition in an organism. And membrane-bound organelle, which is an organelle enclosed in a membrane. So those are the three. Organelle, organized or specialized structures within the cell. We know that we've got and we've seen some of those. Membrane, which is looking at a thin sheet, that membrane-bound organelles, those membrane-bound organelles which don't exist in the prokaryotic cell, whereas we do see them in the eukaryotic cell. And yeah, so that essentially is prokaryotic cells, and make sure you do know examples of each. So you know examples of a eukaryotic cell, you know examples of a prokaryotic cell. So, looking at the comparison between the two, between a prokaryotic and a eukaryotic cell. So, prokaryotic cells, for example, have the similarities in that they both have DNA, right? Prokaryotic and eukaryotic cells, they both have DNA, they both have a cell membrane, and they both have cytoplasm, which is the fluid within the cells. So both of them have those three features: DNA, cell membrane, and cytoplasm. What are the differences? Differences in the prokaryotic versus the eukaryotic. So prokaryotic is more simple, like we initially mentioned. There is no nucleus to contain DNA, so therefore DNA is free floating within the prokaryotic cell. And as we talked about earlier, there is no membrane bound organelles. Okay, when we compare that to the eukaryotic cell is a lot more complex. There is nucleus to contain DNA, unlike the prokaryotic where it's free floating. And finally, unlike a prokaryotic cell which has no membrane bound organelles, a eukaryotic cell does have membrane bound organelles. So those are the key differences between them, whereas the similarities are again DNA, they both have a cytoplasm and they both have cell membranes. So that is how you can differentiate between them. If you were asked in an exam now the difference between a prokaryotic versus a eukaryotic cell, hopefully you would have enough information to be able to identify that difference and distinguish between them. So here's a bit of a summary. So synthesis and processing of proteins and lipids. So nucleus stores genetic material and coordinates the cell's activities. So that's growth, protein synthesis, and cell division. So the nucleus plays a really important role. We've got a ribosome, which acts as the site of protein synthesis, and it's found on the rough endoplasmic reticulum. So remember, there are two types of endoplasmic reticulums, or ERs. We've got the rough one and the smooth one. So the rough ER transports proteins produced by ribosomes, like we initially talked about, into the Golgi apparatus. Whereas the smooth ER is the site of synthesis for phospholipids. Okay? So I know that these might be a lot of new terms for you if you haven't already gone through these, but make sure that you do understand the role of each one. So we've got the Golgi apparatus, which looks at packing proteins into vesicles, modifying them, and then sending them to their destination, whatever that destination may be. Lysosomes digest essentially unwanted matter, such as damaged organelles or foreign material. They're there to sort of digest that and then identify and digest that, essentially. We've got storage and structure of cell structures. Storage and cell structures. So, vacuoles, they provide structural support in plants, specifically. So they're there, again, for that idea of storage and waste disposal. Plastids, which contain genetic material and play a role in the synthesis and storage, so not found in animals. Cell wall, this is a rigid structure that's found in plants, and that provides structure for the cell. So it's in the name, really, cell wall. Cytoskeleton supports the cell structure and movement and assists transportation within the cell. Centrioles are involved in cell division. And finally, the cilia and the flagella are both hair-like structures involved in the movement of cells. So, we can now see that we've identified some of those, so that you can see actually some of those. Smooth endoplasmic reticulum, the ribosome, the lysosome. You can see some of these now that we've been talking about. Rough endoplasmic reticulum, the nucleus, right? And we can see here the cytoplasm, ribosomes, nucleoid, which is essentially DNA, capsule, plasmid, etc. So we can see some of those, and hopefully that's now starting to click. Finally, the energy transformations. A lot of people already know of the mitochondrion. It is the powerhouse of the cell. It plays an important role in the respiration and the conversion of energy from food into energy used by cells. And finally, chloroplasts, which play an important role in the photosynthesis in plants, trapping light energy. You may have already heard of chloroplasts and photosynthesis because you may have gone through it earlier. But yeah, this is essentially sort of broken down in accordance to what their role is, and hopefully that should make it a little bit easier to sort of identify what each one does and what each one means. So yeah, that's essentially the cell organelles and their different functions. So hopefully that's all making sense. Okay, moving on now to organelles and their cellular arrangements. So we have some things that we need to identify here. So this is again a little bit of a summary of what we went through in the last slide, but in a new format. So we'll go through some like more examples of it to hopefully allow you to get an idea of what I'm talking about here. So energy transformations, we know that we've got the mitochondrion, which is the powerhouse of the cell, plays an important role in the cell respiration and converting food and into energy used by cells like we talked about earlier. So again, this is an energy transformation. What is the transformation? We're getting the food, we're converting it into energy that the cells can then use, right? To complete whatever function they are completing. Now, chloroplast, like we talked about on the last slide, plays an important role in photosynthesis because, which is in plants, and the way that they do this is they trap light energy and then use that. So those are energy transformations. Make sure you're aware of what's involved in energy transformations, what those transformations are, and then we can go from there. Okay, so that is the first part. Now let's go through cellular respiration as an example. So, respiration is the process by which cells use oxygen to break down sugar glucose to obtain energy. So, that is going to be our definition of how cellular respiration will occur. Okay, so again, cells use oxygen to break down sugar glucose to obtain energy. Now, there are two ways that this can be done. Aerobic, so aerobic respiration is carried out in the presence of oxygen. So 36 ATP are produced as a result, versus anaerobic. So the way that you can remember it is an means no oxygen because respiration is carried out in the absence of oxygen where 2 ATP are produced. So aerobic is with oxygen, anaerobic is without oxygen, and aerobic produces 36 ATP, and aerobic produces only 2 ATP. So making sure you're understanding those differences, making sure you're understanding how respiration occurs. Again, looking at the use of oxygen to break down sugar. Okay. Then we have photosynthesis, what we were talking about with chloroplasts, even in the previous slide. So photosynthesis is the process by which plants use light energy, like we talked about. They trap the light energy to produce their own food in the form of glucose. So light energy transforms to glucose, which is their form of energy. So this is carried out in two specific stages, like we had with aerobic and anaerobic in cellular respiration. In photosynthesis, this is carried out in two stages, which is the light dependent and the light independent stage. So the light dependent stage is photolysis, which occurs in the grana. So it's really important to be able to identify where it occurs as well and the two types. So make sure you are able to correctly identify that. So light dependent stage occurs in the grana. Light independent stage, which is also known as carbon fixation, it occurs in the stroma. Okay, so you want to be able to identify where they sit in the actual cell. So again, a little bit of a summary of this slide because I really want you all to be able to really understand in detail what I'm talking about here. Mitochondrion and the chloroplast are the two energy transformations. What is the one for mitochondrion? They're converting energy from food into energy used by cells, and that's how respiration occurs. Cellular respiration. We have cellular respiration, which is the use of oxygen to break down sugar, also known as glucose, to obtain energy. How is this done? It's done by aerobic and anaerobic. So aerobic produces 36 ATP, anaerobic produces 2 ATP. Photosynthesis, we've got the process by which plants use light energy to produce their own food. What's their food? Glucose. So that's the form. Again, it's carried out in two stages, the light dependent and the light independent stage. What's the light dependent stage? Also called photolysis. Independent called carbon fixation. Where does the light dependent stage occur? In the grana. Where does the light independent stage occur? In the stroma. Okay, so that's a quick summary again of the organelles and their cellular arrangements, and we've gone through examples now of cellular respiration and photosynthesis. So, next we're going to investigate the way in which materials can move into and out of cells, including but not limited to conducting a practical investigation, modeling diffusion and osmosis. Again, think back to how to read the syllabus. You need to be able to identify what's involved, be able to identify the terms. If I go back over here, so read the working scientifically skills. Then you want to take note of the inquiry question and pay attention to the wording. These 3 steps are so important to success in this course, right? You want to be able to identify how to accurately read the syllabus, how to correctly read it, and how to correctly identify and approach it when you get to the stage where you want to be able to write exam answers that conform with the syllabus. So, passive movement of materials. So there are 3 types, right? Diffusion, osmosis, and the concentration gradient. Make sure that you know these and you know how they work. So diffusion is the passive movement of materials from high to low concentration until an equilibrium is reached. Osmosis is the passive movement. These are all going to be passive movements. Give me a sec. So diffusion again, high to low concentration until equilibrium is reached. Particles. Osmosis is the passive movement again of water from high to low concentration across a semi-permeable membrane until equilibrium is reached. And finally, concentration gradient, which I have got the diagram over here, is the passive movement of water from a high to low concentration across a semi-permeable membrane. So you can see the differences between them, right? So you want to be able to identify the differences between diffusion, osmosis, and the concentration gradient. So we've got the example of concentration gradient here. So remember that it's the movement of water from high, so over here, all the way to low concentration across a semi-permeable membrane. Osmosis, we have 3 types of solutions: the hypotonic solution, the isotonic solution, and the hypertonic solution, right? So here's the diagram as well. So we've got the different areas conducted for osmosis. So again, be able to identify diffusion. What is it? It's a passive movement of particles from high to low concentration until equilibrium is reached. Osmosis, passive movement of water from high to low concentration across a semi-permeable membrane. Again, until equilibrium is reached. And concentration gradient, passive movement of water from high to low concentration across a semi-permeable membrane. Those are the 3 for passive movements that you want to be able to remember. Moving on to active movement of materials, so active transport. The process requires ATP, which is energy, like we talked about earlier in respiration. So needed to move against the concentration gradient, which we have over here. So we need to now move against it, which is going to be a little bit different to passive. So may be needed if a substance is actually charged or it's too big to be able to originally diffuse, which is a process of diffusion, right? So they can't actually undergo that process. So then we have active transport that occurs, and there are 2 types: endocytosis and exocytosis, and the way that you can remember them is through the names, which I'll talk about in a second. So before we get into that, how does endocytosis occur? It's when particles are too large, membrane changes its shape so that it can surround and engulf a particle and take it into a cell. Now this is known, or an example of this rather, is phagocytosis. So again, it will change its shape, surround and engulf. Okay? Exocytosis, however, is a little bit different. It is where membrane-bound vesicles move to the cell membrane, fuse with it, and then release the contents into the environment that's surrounding it. So transport substances out of the cell that are needed elsewhere or are in too high of a quantity in the cell. Okay? So again, one of them is moving in, one of them is releasing, or it's going out. So endo in, exo out. Okay? That's how you want to remember it. That's how I remembered it. So you can see the diagram over here. See the extracellular fluid. You can see the inside of the cell. that's where we see active transport occur. And there's a diagram example there as well. Right, next up, we've got conducting a practical investigation to model the action of enzymes in cells. So, enzymes are highly specific biological catalysts that bind with specific substrates called the activation site, right? So, the way that we will look at this is through the lock and key model, right? So, we can see those steps. I won't go through each step. Actually, I might just go through it. It's actually fun. So we've got this. The bond substrate is sucrose, which is composed of glucose and fructose that's bonded together. So the substrate will then bind to the enzyme, and you can see it's a lock and key model. And that's where the name comes from, as you can see where it fits into the lock and key. Number 3, bond binding, the substrate breaks. Number 4, the products are released. So you've got the fructose, you've got the glucose. And number 5, the enzyme is free to then bind with other substrates. Okay? So you can see that enzymes movement, and then you can also see the movement of the substrate. Yeah. This is a little bit different from the induced fit model, where first you have the substrate again of sucrose, which is composed of glucose and fructose, that stays the same. So substrate binds with the enzyme, but it's an induced fit. So the binding of the substrate induces the enzyme to change the shape to fit more tightly, rather than the lock and key. And then the steps are the same after that. Okay, so that's the difference. Make sure you understand the difference between a lock and key and induced fit. You can see the difference. Like induced is like it's sort of changing around to fit, whereas a lock and key is just sort of happening immediately. That sort of concludes our overview of Module 1. So it wasn't in too much detail, but I just wanted to still have that in there for you all to access and to provide a bit of a summary of it. We'll now move on.